Can you hear me? Yeah, wow. A lot of volume. Better than the other microphone, I think. A couple announcements. One thing I do is I put up on the board over here every lecture, office hours, remind you of the office hours. First, I actually was down in 2084 on Wednesday, and there were a few students there. It surprised me, but that's okay. So 2084 from 9 to 10 today, I'll have office hours. And if, as we go on, it's a small room. Last year when I taught, there were lots of students coming to office hours, which is rather unusual. It's nice, but unusual. So we found a classroom nearby that we could go in. It was empty, so that was good, because the room only holds 15 people maybe. And then I put up also something related to the lecture that I'm going to give today. Some of the topics that I'm going to cover, I don't necessarily get through everything on the board, okay? But this is for those that come in late and don't know where I am, or those that fall asleep and don't know where I am, things like that. Mike Meehan wanted me to announce that if you want to request a section switch, you have to go to BSpace and submit your request between Saturday noon and Sunday noon. Now, switching sections is very difficult in this class because most sections are full. And there's not space in another section. We can't put more people in sections. Sections are driven by lab space. So this doesn't mean you're going to get your switch. But if you want to request a switch, that's how you do it. The other thing is, I looked after lecture yesterday in the afternoon for the webcast on the Berkeley website for the webcast, and it wasn't there. I don't know if it's there now. But the university has something going with YouTube. Are you aware of this? There's a Berkeley YouTube channel. And the lecture was on the Berkeley YouTube channel when it was not on the Berkeley webcast site. So, you know, maybe you have to Google Berkeley YouTube channel or something, and you'll find it. And they seem to, I don't know why it gets up there quicker than the webcast portion. But it was there, and actually the quality was pretty good. Sometimes the quality of these webcasts isn't very good. The other thing I would say about the webcast, for those people at home who don't want to come in, sometimes there are technical problems. I'd say maybe one lecture out of 13 or 14, the sound doesn't work or the camera freezes, and the webcast is not available or not available for the whole lecture. And you know, people are going to come in and ask me, well, can we do something about it? No, we can't do anything about it. You know, when there's a technical problem, it's gone. So it's another, I think, very compelling reason for why you want to come to lecture. Okay, I'm going to continue to talk about protein structure. I started talking about protein structure on Monday mostly in relation to amino acids. I talked about amino acids and the different kinds of amino acids that are found in proteins and how you can organize them according to the R group, the chemistry of the particular R group. And I started to talk about the different levels of protein which we consider in relation to protein structure. And I've put the summary on the boards here, and I'll go through this. The first one was, of course, the primary structure of a protein is its linear sequence of amino acids. So every protein has a specific sequence of amino acids, and every protein has a different sequence of amino acids. The secondary structure are organized local regions. I brought in my toys today, okay? I've got a protein here. You can see this protein. Every pink thing is a different amino acid, and the way you build a protein out of this thing is, if I can do it, there's an amino acid, there's a polypeptide, okay? What happens is there are regions, and using wire might be a little better, there are regions in proteins which are organized by peptide bond hydrogen bonds. And they can form alpha helices or beta sheets. So here's a little helix, and this is supposed to show you something about a beta sheet. And this is not the total three-dimensional structure of the protein. This is a small portion of the protein which can assume this particular structure. And somebody asked me already, well, why? What makes an alpha helix and what makes a beta sheet? Actually, the amino acid side chains, because they're sort of sticking out, and their ability to form alpha helices or beta sheets is driven by the spatial arrangement. Although the side chains are not stabilizing these two structures, the alpha helix or the beta sheet. They're stabilized by hydrogen bonds, which is shown in figure 11. You've got hydrogen bonds going from one sort of screw of the helix to another in the alpha helix. And in the beta sheet, you've got hydrogen bonds going between sort of parallel or anti-parallel portions of the protein. But the important thing that you have to remember is these are local regions. This is not the entire shape of the protein. Tertiary structure, as I've written here, is the final or global three-dimensional structure of a protein containing a single polypeptide. So this is the tertiary structure of this protein. And this protein, or this protein, I won't have any, I'll just fold it. I love this because you can just make a protein. There's a folded up protein. The protein folds up like a snake, okay, into its tertiary structure when there is one polypeptide. There's only one polypeptide in this protein. The tertiary structure of a protein is stabilized by R group interactions. Side chains of amino acids are interacting with each other to stabilize these proteins. And figure 10 gives you the 3 or 4, depends on you count, major ways that tertiary structure is stabilized. And these are very important. One is called disulfide bonds. I'll talk about these. Disulfides, which are SS bonds. This is covalent. Covalent, meaning these are very strong bonds, hard to break. 2 hydrogen bonds. 3 ionic bonds. And 4 hydrophobic interactions. They're not exactly called bonds. All of these interactions are relatively weak. And hydrogen bonds can be formed from NH2 and carboxyl groups or things like that. Ionic bonds are from positively and negatively charged amino acid side chains. Hydrophobic interactions mean you take the hydrophobic amino acid side chains, leucine, valine, and they will interact to stabilize tertiary structure. The most interesting type of stabilization in tertiary structure comes from what are called disulfides. If you go back and look at, when we finish, at the list of amino acids, there is an amino acid known as cysteine, which is an R, that's the whole, well, I won't, okay, you have a CH2 and you have an SH group in that. That's a sulfhydryl group. Sulfhydryl groups can oxidize to form an amino acid known as cysteine, where you have CH2, S, CH2, okay, like that. That is a very important stabilizing factor of tertiary structure, because this is not a weak bond. This is a strong covalent bond. It can be reduced under certain conditions, but it really puts a lock in the structure. Like, I mean, I could conceivably have a disulfide bond across there, and that would lock that region of the protein in its particular structure. So we have tertiary structure stabilized by R group interactions, secondary structure stabilized by peptide hydrogen bond interactions, and I guess you would say primary structure stabilized by what? Peptide bonds, I mean. as I said, the final three-dimensional structure of a protein with a single polypeptide chain is its tertiary structure. Every protein, I'll come to this in a minute, every protein has a unique three-dimensional structure. It's also called a conformation. And that structure is the biologically active form of the molecule. This structure is established by the folding of a protein. So as a protein is synthesized in the cell, it doesn't end up as a long linear chain for various reasons that we could go on to. As it's being synthesized, it starts to fold up into a structure which is its tertiary structure for many, many proteins, and that structure is its active form. Now, if I change the structure, I change the function. That's one of the main messages of all of my lectures. Structure and function are related. If you look at figure 13, because I'm not going to draw this, what it is it's the amino acid sequence, the linear structure of insulin. Insulin is a small protein. I think it has a molecular weight about 6,000. And you can see there are a bunch of amino acids at the top of this figure, and then below it is a second bunch of amino acids. That's because insulin is a protein that has more than one protein polypeptide. Proteins that contain more than one polypeptide have quaternary structure. Lots of proteins have more than one polypeptide. And of course, what you have in this case is you've got often two different polypeptides associated with each other to fold into the native conformation of that particular protein. You will see the term subunits used very commonly in the literature. So people say, well, this protein has 8 subunits. Protein has 4 subunits. That means it has 4 polypeptide chains. And as I said, they can be the same, or they more usually are different. So some proteins have their tertiary structure. Some proteins end their structural manipulation with a tertiary structure because they only have a single polypeptide. Others have a quaternary structure where you have more than one polypeptide. Proteins in solution, and we're going to actually be talking about proteins which are not in solution but are actually in the membranes of cells, are called soluble proteins. And these proteins assume what everybody calls, again, a term you might see in the literature. If I can get this on and get this working. There, great. Okay. That picture off to the right is the structure of a protein known as myoglobin. It's a protein that carries oxygen in your muscle. And what you see in that particular figure is what's called the backbone, the peptide portion. You don't see every amino acid. You don't see every side chain of every amino acid. And I think the reason myoglobin is kind of cute is you can see this is a protein that primarily has alpha helices in it. There's no beta sheet. But if you look at the actual complete structure of this protein, this is what it looks like. I mean, these are all the amino acids, side chains, and you can see it's a glob. That's where the term globular protein came from. We're going to talk a bit more about myoglobin later. How do proteins fold? That's one of the big questions that bothered biochemists for a long time. A very, very clever experiment was done, I think it was done when I was a graduate student in the 60s, by a researcher at National Institutes of Health in Bethesda, Anfinson. He took an enzyme, doesn't matter what it was, but it was ribonuclease. Ribonuclease is a protein that has 4 of these disulfides. It has 8 sulfurs, and they form 4 couples of disulfides. And I've labeled them 1, 2, 3, 4, 5, 6, 7, 8. This is also shown in figure 15, but I've put in the sulfhydryl groups. What Anfinsen did was he denatured the protein. What does that mean, denature the protein? He got the protein to unfold, and the way he did that was actually by adding some reducing agents and reduced these disulfides to SHs. I'll put H's on here. And proteins can be unfolded or denatured, and the effect of denaturing a protein on activity, what do you think that would be? Would this protein be biologically active? No, it's totally inactive. So you go from 100% activity to 0% activity, okay? And then you remove whatever agent you've added to unfold the protein, and the protein refolds. And when the protein refolds, it refolds into its native conformation. Now, maybe this doesn't surprise you, but you've got all of these sulfhydryl, these SH groups, and there are probably, I don't remember what the number, there are many possible combinations. Like this one could react with this one could react with that, this one could react with that, this one could react with that, something like that. It doesn't look right, okay? But you can see there are a lot of possible arrangements for disulfides to form. But the only disulfides that form are these ones, 1 with 8, 2 with 7, 3 with 6, 4 with 5. And 100% of the activity is restored. And the reason this was a very important experiment, it said that the information for a protein to fold into its native conformation is in the amino acid sequence of that protein. If you lay down this amino acid sequence, you're always going to get this particular conformation. And in fact, this was done when people made machines that actually synthesized synthesized proteins. You could feed in information, make me a protein that has this sequence, and the synthesizer would make the protein, and then you take the protein out of the machine, and you let it fold. It would fold into the native conformation. Nothing else was required. And that was a very dramatic demonstration that protein folding is somehow, the information for folding is somehow embedded in the linear sequence of the amino acids. Protein folding is very important. If a protein is misfolded, it doesn't work. I found an article a couple years ago in the New York Times. They listed about 6 or 7 diseases which are protein folding diseases, including Alzheimer's, diabetes, things like that. These are diseases that are related to misfolded proteins. So the important point is you have to have a system built in that properly folds proteins. It turns out that what I just said, that all of the information for folding is in the sequence, it's true for many proteins, but it was also discovered more recently that there are proteins which are known as chaperones. You know what a chaperone is, right? Not anymore. In the old days, in the Victorian times, you know, when a couple went out for a walk, they didn't go by themselves. They had somebody who came to make sure there was no hanky-panky or anything. Now, chaperone. So there are actually proteins that assist folding. And I'm not going to go into the details of this, but what I'm struck with is folding is so important that there is actually a group of proteins that assist in the folding. They are, for the most part, involved with proteins that have a quaternary structure, where you have to bring, say, 2 different kinds of subunits together. These proteins they function sort of by acting as a scaffold to bring subunits together and allow them to fold properly. So protein folding in the cell is so important that there are proteins that help fold proteins. The amino acid sequence of proteins. As I showed you, insulin was actually the first protein for which an amino acid sequence was solved, and this was solved by British biochemist Fred Sanger in probably the 50s. And the way proteins were sequenced in those days was chemical. In other words, you had a protein, let's say you had 50 amino acids. You chopped off one amino acid at a time, and you identified it, and then you kept going on and kept going on and kept going on. It was a very, very long, tedious job to do this. Things improved substantially when molecular biology came on the scene. A lot of proteins were sequenced by just chemical methods. But once we understood the relationship between DNA and RNA and protein, people no longer do chemical analyses of proteins, sequence analysis. They sequence the gene, and that gives them the sequence of the protein, and you'll learn all about that in the second portion of the class. Once you have proteins isolated, purified, you can do some interesting things with them. One of the things that was done in the 1930s with an enzyme called urease was it was found that one could crystallize proteins. You know, if you take organic chemistry, inorganic chemistry, one of the objects of isolating a complex is to get a crystallized form of it, then it's very pure. And it wasn't believed you could do this with proteins because proteins are very high molecular weight, hard you'd think, to crystallize. And urease was crystallized in, I think, 1932. It was the first protein that was crystallized. And subsequently, lots and lots of other proteins were crystallized. But what people discovered when they could crystallize proteins was that it was possible to obtain the complete three-dimensional structure of a protein using a technique called X-ray crystallography. You take the protein crystal, it's got to be really pure, and you put it in an X-ray beam, and the beam comes in, and the organization of the crystal allows the X-rays to be diffracted, and you have a piece of X-ray film, and you end up with a bunch of spots on the film, and I'll show you the famous film for DNA later, done the same way. And if you're very smart, much smarter than me, you can take all these spots and you can convert them into the three-dimensional structure of that protein. And if you're interested in the function of a protein, you have to know its three-dimensional structure. So two British biophysicists working in the 50s and 60s. One was, and the names I mention you don't have to worry about, okay? I like to mention names of people because I like you to realize that well, everything we talk about in this class, somebody went into a lab and did an experiment. I mean, you know, wasn't just pulled out of the ether. So two British biophysicists, John Kendrew and Max Perutz, working in Cambridge University in the UK, were working on myoglobin, that's protein I just talked about, and hemoglobin, sort of myoglobin's brother, which is the oxygen-carrying protein in our red blood cells. They crystallized these proteins, and they deduced or worked out the first three-dimensional structure of proteins. And I had the myoglobin one up here. This is where this kind of figure comes from. So this represents the polypeptide backbone in myoglobin. Myoglobin is a lot simpler than hemoglobin. Myoglobin has a single polypeptide subunit, so this is its three-dimensional structure. If you look at figure 18, no, it's not 18. 8B, no, it is 18B, I'm sorry. Hemoglobin is much more complicated. It has 4 polypeptide subunits. So hemoglobin is a protein that has quaternary structure. And there are 2 kinds of subunits in hemoglobin, alphas and betas. Somebody asked me about the nomenclature that's used in biochemistry, alpha and beta. Does this mean anything? It means absolutely nothing. It could have been called, you know, George and Martha. So hemoglobin is roughly 4 times bigger than myoglobin. It's a very complicated protein. It contains something which is shown in C. To the right, it contains a component which is known as a heme group. So hemoglobin and myoglobin, MB, contains something known as heme, and we'll be talking about heme later. Heme is something that is attached to the protein for a specific function. Heme contains iron, and that's where the iron is actually bound on hemoglobin and myoglobin. The other thing that's come out from three-dimensional structures, and there are probably thousands of three-dimensional structures that are now available. Obtaining the three-dimensional structure of a soluble protein, you need a good crystal and you need a lot of it. Hemoglobin is great because it's readily available. All you've got to do is bleed a little bit. So you start to get a lot of information on protein structure. And if you look at figure 17, I put figure 17 in only to show you representative protein structures. The first one has a lot of alpha helix. Doesn't even matter what these proteins are. The middle one, all of those arrows are beta sheets, a lot of beta sheets. And the third protein on the right has a combination of alpha helix and beta sheet. So you can have all kinds of mix and match possibilities. The other thing that's come out from these detailed structures is shown in the first part of figure 18. Next to hemoglobin, you have proteins. Let's see if I can use my protein here. I'll do that. I'll change it a little bit. You have domains within proteins. So here's a domain, and here's a domain. These are regions that kind of stick out and say, look at that region at the top of the protein is different than that region at the bottom of the protein. These domains are for differing types of interactions with different molecules. In the case of enzymes, which we'll talk about in some detail, you have various enzymes, a group of enzymes that work with, let's say, ATP. Everybody must know what ATP is. If you don't know, you'll know a lot about it. Just the compound, okay? So an enzyme, groups of enzymes that work with use ATP for various purposes will generally have the same interacting domain. It won't be identical. So there may be this top region, which is an ATP binding domain, and you would find that if you were working with a protein, you didn't know what it did, and you could get a structure, and you found a domain that looked like the one on the top, you'd say, well, this is an ATP binding domain, and you'd probably be right. And then you'd say, well, what does it do in relation to ATP? And you could hopefully figure that out. So the structural information that we have gotten over, as I said at the beginning, over the course of the last 30 or 40 years is amazing. I mean, hemoglobin is probably the most studied protein in the world. We know where every atom in the hemoglobin molecule is. We know there are lots of variants of hemoglobin where you have amino acid changes, and these are related to specific illnesses. The classic example of this is something that probably everybody has heard about, sickle cell anemia. If you look at red blood cells with a person who has sickle cell anemia, it's a bad picture. Oh, it's a terrible picture. I tried to get it better. The last figure of 18 on page 9A. The picture at the right are red blood cells in a person with sickle cell anemia. They look kind of like this versus, these nice round donut-shaped things. These are normal red blood cells. And actually it was Linus Pauling, again, who predicted back in the 1940s, before any sequences had been done and not much work had been done on this problem at all, he said that sickle cell anemia, sickle cell, was a, quote, molecular disease. What did he mean? I don't know what he meant. But he had this idea that the molecule, hemoglobin, was different in sickle cell hemoglobin than in normal hemoglobin. And when people started to sequence proteins, it was discovered, put this up, that in normal hemoglobin, HB, you had a sequence which was valine, and This also is not something you have to write down and memorize, please. Leucine, threonine, proline, glutamic. I'll stop there. This was part of the beta chain in hemoglobin. In sickle cell hemoglobin, this was the same, this was the same, this was the same, this was the same, this was the same. This was valine. So what you've done in sickle cell hemoglobin is you have replaced a charged amino acid side chain, negatively charged at neutral pH, with a valine, which is a very hydrophobic amino acid. The effect of this is for sickle cell hemoglobin to clump up and make patches within its red blood cell, producing this sickle cell shape, and also clearly lowering the affinity of the protein for oxygen. So people who have sickle cell anemia usually do not have a very long lifespan. And this is the best example, simplest example I can give you of how the change in the structure of a protein alters its function. I'm sure that the three-dimensional structure, a very high-resolution structure of sickle cell hemoglobin is available, and all that I have said has been confirmed by this. Okay, that's all I'm going to say about proteins. We'll talk about proteins again and again and again. We'll talk about them in relation to enzyme structure and function, and we will also spend a great deal of time not talking about soluble proteins, but proteins in membranes, which carry out a series of very important reactions in most cells, respiration, photosynthesis, for example. Okay, the next group of molecules that I want to talk about are lipids, and I always find lipids to be dull. They're really dull. But obviously they're important, because they kind of sit there. Proteins are active. They're binding antigens, and they're carrying oxygen around and transferring electrons, and lipids kind of sit there. Okay, what are lipids? Lipids are a very diverse group of molecules that have one thing in common, and that is they are insoluble in water. So the first thing you should think about when you think about lipids, or are asked anything about lipids, are these are hydrophobic molecules. And there are several different kinds of lipids, diverse group of molecules. One are fats, two are phospholipids, which are very important, and three are, as an example, steroids. Okay, fats. Here's where I hope you have your book, because these structures are not simple to draw. They're simple in concept, but if I draw them on the board, it would take me 15 minutes. Fats are found in things called fatty acids. And fatty acids contain two different elements. If you look at them, I'm looking at this figure and I'm so unhappy with it. Okay. Fatty acids come in various varieties. Okay. They're long-chain carbon-containing compounds. So if you look at common fatty acids, you can see they have 16 or 18 carbon CH2 groups with a carboxylic acid at one end. I think when you look at these particular structures, you should immediately realize why they're hydrophobic, why they're insoluble in water. There's no the single little negative charge in the carboxyl group is overwhelmed by 16 carbons, okay? So this is a very hydrophobic molecule. The difference between C18 stearic acid and C18 oleic acid. There is a double bond in oleic acid right in the middle of the molecule. This is an unsaturated fatty acid because 2 hydrogens have been removed. And the effect of the removal of those 2 hydrogens is shown in the molecular model versus one versus the other. You get a kink in the molecule. So this would be a saturated, this is the way we draw these. Saturated fatty acid looks like that, and an unsaturated one looks like that. Okay, and the double bond is here, and it forms a structure, a different structure than a saturated fatty acid. Now, to get fats, what you have to do is you have to combine fatty acids with a molecule called glycerol. Glycerol is a simple molecule. It's an alcohol. 3-carbon alcohol, like this. That is glycerol. And what happens in the formation of fats is this OH reacts with this carboxyl, and we've got all of these carbons sitting out here to form an ester. There's an ester bond between the acid and the alcohol. That's what's shown in figure 20. You have 3 OH groups. You can have a mono, the names of these are as follows: monoglyceride, triglyceride, or diglyceride. Whether there's 1 fatty acid side chain, 2 fatty acid side chains, or 3 fatty acid side chains. This thing is really awkward. So the figure in figure 20 is for a triglyceride, also known as a triacylglycerol. I don't use that term, but you'll see that. And the way you get these is you have this condensation reaction where this OH and this go away as water, and there's a bond that forms there, okay? And I think that's what's shown in there. Yes, it is. So you have these 3 different kinds of fatty acids. Fats are have become a topic of great discussion, let's say, okay? You cannot pick up a newspaper without somebody writing an article about what you should eat and what you should not eat. And one thing you should not eat are foods that have lots and lots of saturated fatty acids. The classic example of a saturated fatty acid is lard, for example. And, you know, animal fats. Animal fats are now considered to be bad for your heart. I think there's no doubt about it that this is true. Plant fats, which turn out to be oils, like olive oil, sits at the top of the chain of great things. Olive oil, canola oil turn out to be liquids. Plant fats have lots of unsaturated fatty acids. The difference between something being a solid or a liquid is related to the fact that these unsaturated fatty acids, what they say, pack differently than saturated fatty acids. How they immediately affect your arteries and your heart is a different issue. So the 2 kinds of fatty acids that exist in biological systems, the saturated versus unsaturated, are related, I think, in a rather dramatic way with heart disease. And when you go out and you eat one of those greasy hamburgers, just think what you're doing to your arteries, okay? Okay. I won't talk about fats anymore because they're involved in other things, but they're not relevant to pertinent to what we're talking about this particular portion of the class. A group of molecules that is very important in terms of what we're talking about are molecules called as phospholipids. You have to look at figure 21 and also 22. Figure 21 is a phospholipid, and what you see in this molecule it isn't shown in detail, but there's a glycerol backbone, so that's glycerol. And one of these glycerols has a fatty acid on it. Another one has a fatty acid on it. Okay, so this is glycerol. And you can see that in the figure, the CH2, CH2, CH2 are the glycerol portion. The fatty acids sticking down are hydrophobic fatty acids. Those are the two I've written here. But the third oxygen in the glycerol is not esterified to a fatty acid. It is esterified to a phosphate group. I'll do it this way. That is a phosphate group. A phosphate group comes from, this is phosphoric acid. And this is PO4 minus, minus, minus. That is a phosphate. That's the anionic form of phosphoric acid. So you have in this molecule a phosphate group, which makes it a little different, certainly makes it different than a triglyceride, where you've got 3 fatty acids. But also, in addition to that, one of these O's is esterified to another polar group. What do I mean by a polar group? Well, what's shown next to figure 21 is figure 22. That's clever. And in that, it shows the kinds of groups that you find here. The most common one, or the one that's talked about the most as an example in books and things, is choline, which is in that particular figure 22. This compound would be phosphatidylcholine if this were a choline molecule. The reason I give you figure 22 is so that you will realize there are other kinds of compounds that can be here. For example, you might have an amino acid such as serine. That's a polar group. That's a polar compound. Ethanolamine, even glycerol. So you can have the glycerol backbone and you can have glycerol here as well. But in each case, this is a polar group which is attached to this very hydrophobic general molecule. So these molecules, which are phospholipids, they have 2 identities. They have the head group. This is called the head group region. Head group, which is polar. Why is it polar? It's polar because of this, and it's polar because this oxygen also might be ionized. So you've got a charge from this oxygen, and you've got charges, or usually let's say charged groups in your polar group. So this is a very polar portion of the molecule. This, of course, is very nonpolar, hydrophobic. Okay, so that's a phospholipid. When you take these molecules and you throw them into water, something rather amazing happens. You end up with something which is known as a bilayer, a phospholipid bilayer. Phospholipid bilayer. And what that looks like is this. This is a schematic drawing. Looks like tadpoles. And I can go on and on and on drawing this. I forgot something I wanted to say about globular proteins. I'll come back to it. This reminded me of it. Okay, what is that? That's the structure of a phospholipid bilayer. These are the head groups. So there is water out here and water here, and this is the hydrophobic core. Which is totally non-aqueous. No water in there because that is where the nonpolar portion of the molecule is. The interaction here is not covalent. These are not covalent bond interactions. The primary interaction in this bilayer, as it's called, one-layer layer, is the hydrophobic interactions between all of these fatty acid side chains. Okay? And if you've got a lot of these side chains, these interactions are very, very strong. So this is a very stable structure. The binding of this phosphate group in a phospholipid, I'm going to introduce this term now, and then we're going to come back to it several times again. This is known as a phosphoester bond. It's a phosphoester because you've got an OH here and a phosphate group here. The acid, an ester is the combination of an acid and an alcohol. The alcohol comes from glycerol. The acid comes from phosphoric acid. So this is known as a phosphoester bond. Actually, it is known as a phosphodiester bond because one phosphate is esterified here, or the phosphate is esterified here, and the polar group is esterified here. This is a structure that comes up again in nucleic acids, and it comes up in when we talk about other phosphate-containing compounds, such as ATP. So I want you to start to recognize these different kinds of phosphate bonds. They're very, very important in biology. Why are phospholipid bilayers? Why do I spend all this time on these? Why are they important? Well, this is the basis of biological membranes. This is the structure of a biological membrane with some modifications that you will see later on, probably the end of next week. Biological membranes are built up of phospholipid type bilayers. Okay, very briefly, the third group of steroids that I want to talk, lipids I want to talk about are compounds which are steroids, and they're shown in figure 24. And again, here you see molecules that basically have different structure. They don't look at all like fats or phospholipids, but what they have in common is they're insoluble in water. Hormones, the molecule cholesterol, very, very hydrophobic molecules and should be considered as lipids. Carotenoids in plants, which you find in the fall, the reds and the yellows, are very hydrophobic molecules, and they are considered as lipids by me. I don't know if everybody else does. And chlorophyll, which is also, you see it as a long tail. I'm looking at figure 24. A long hydrophobic tail of carbons, very hydrophobic, can be classified as a lipid. I know what I forgot to tell you. It's about proteins. I'm going back because it's important. I talked about globular proteins, and I showed you the structure of myoglobin. So a globular protein, you write this down because it's important. The reason it forms this globular structure is if you look at the actual structure of these proteins, you see the surface is filled with charged amino acid side chains, which can interact with water. So they're on the outside of the molecule. The inside of the molecule has all the hydrophobic amino acid side chains. They're away from the water, and this is a relatively stable conformation. It takes energy to put a hydrophobic amino acid side chain into water. So what drives this globular formation of structure in most soluble proteins is the polarity of the amino acid side chains. And this is what I sort of keep talking about over and over again. Okay, so I am not going to talk about carbohydrates at 2 minutes to 9. I'll talk about that on Monday very briefly, and also very, very briefly make a few comments about nucleic acids, okay? So everybody should hope for the 49ers this weekend. That's the important thing.